everyone, my name is Jeffrey Perera, and today I'm here at the Gladstone Institute with Dr. Alexander Marson. He is the director of the Institute of Genomic Immunology and is also a professor of medicine, microbiology, and immunology here at UCSF. He leads a highly translational lab that focuses on the genetic circuits that control the immune system, especially in T cells. On top of all this, he is a scientific founder at Arsenal Biosciences. Alex, it is a pleasure for me to be speaking with you and to be given the opportunity to hear your story firsthand.
Great to see you.
So, I've introduced you a little bit, but I'd want to hear it from you. Who are you, and what do you do?
I am a scientist. I was trained originally as an infectious disease doctor, and that's also part of my identity, but I don't see patients anymore. Now I run a lab that's focused on understanding how the cells in our immune system are controlled by the DNA inside of those cells. And I get to work with an amazing group of people that stretches from people in high school, in college, all the way up to people who have made a career in science. And we get to work together to ask some really hard questions and discover things that have never been known before. And I think that that's the addictive part of what I get to do. Both of really three things. I get to work with incredibly smart people. There's something absolutely motivating about the ability to push the boundaries of what we understand, and then to try to use that understanding to try to help patients who really need new and improved treatments.
So, take me back to your earlier years, from your childhood to studying as an undergrad at Harvard. When you think back, what is your earliest memory of falling in love with the sciences?
I think in some ways, where I really think that passion was first came to the surface for me was actually in when I was in grade school. My school had an archaeology course, and I think that's where I really got introduced to the idea of discovery. It was a fake archaeology site. They had a big tub of dirt and they buried archaeological artifacts in there, and we had to excavate and we had to make some deductions about where the site was from and why the artifacts were arrayed the way they were. And I think that turned me on to the idea that there were these opportunities in life where there were hidden facts, and that if we looked in the right place and used the power of our minds, we could discover things. And that's not the kind of discovery we do anymore, but that is very much the same joy that we get to find in biology.
Could you have seen yourself doing anything outside of a career in science?
Absolutely. I think there are people who have a very clear sense that science is exactly what they're going to do. I don't think that I was like that. I think I was curious about the world, and that curiosity progressed into different directions at different times. And science became one amazing outlet that I love, and I feel really privileged to get to work on every day. But I don't think it had to be that way. I actually thought pretty seriously about having a career as a chef. I was thinking pretty seriously about going to culinary school at one point, and I don't know what that life would have been taken, but I imagine I could have been pretty happy and enjoyed that too. And I suspect that there are other things that I would have enjoyed, but I feel very lucky that it turned out the way it did.
As teenagers, we're often told to quote, "You're the average of the 5 people you spend most time with." What were your friends growing up like, and to what extent do you agree with this statement?
Yeah, I think I think friends are incredibly important in shaping our ideas, and it's another area that I feel really fortunate with. I've had the benefit of really long and really great friendships in my life. My friend who I announced when I was 3 years old was my best friend, is still my best friend, and there are a number of other people that I've known now. I guess it's funny, it sneaks up. It's who I've now known for decades, and of all the things in my life, one of the things I'm most proud of is the group of friends that I've managed to accumulate, and I've certainly been the beneficiary of having incredibly thoughtful, fun, loyal friends, which opens up a lot of possibilities and also makes you feel grounded to take new risks. I think I think one of the things is about old friends is they keep you honest and give you some accountability over time and a reminder of who you are. And also then that in turn can give the freedom to explore new ideas and feel safe that you can go back and have sounding board of people you really trust over time.
Many of the keywords throughout your research are CRISPR and CAR T cells. In the context of the human immune system, how can CRISPR be used in the study of T cells, and why are T cells such an important candidate of study in disease?
So T cells are a type of cell, they're a type of white blood cell that is found in our blood, and we've known for decades that T cells are a critical component of how we organize an immune response. They are critical for protecting against types of infections. They really are orchestrators of our immune response. And just over the past decade or so, it's really become clear that in addition to the ways that they naturally protect us, those cells can be engineered to offer us extra protection or treatment for a disease. And where that's gone the farthest is CAR T cells. So CAR T cells have been really a remarkable advance in opportunities of how we treat certain types of cancer. And the way that CAR T cells work are that T cells are actually taken out of a blood sample. So it's like giving a blood donation. The T cells are taken outside of the body, and then they're genetically modified to transform them into CAR T cells. The CAR stands for chimeric antigen receptor. And that's an artificial receptor that doesn't exist in nature, but when this gene that puts the CAR into the DNA of the T cell goes in, it makes an artificial receptor on the surface of those T cells, a little sensor that is found on the surface of these cells that allows the cells, when they get reinfused into a patient, to survey throughout the body and sense where they can find cancer targets. And there are now patients alive and really apparently cured in a long-lasting way from CAR T cells who would have died without CAR T cells. So I think that has taught me and taught the world that this is not a fantasy, that genetically engineered T cell therapies can cure disease. Now, we know that this still doesn't work for most patients with cancer. It only works for a subset of certain types of blood cancers. We want to make those responses last longer so that patients can really be long-lastingly cured. And we want to take those successes and extend them to other types of cancer, to tumors that still don't respond to CAR T cells. And that's where CRISPR comes in. That CRISPR gives us much more power, much more precision, much more flexibility of how we do these types of genetic edits to really be able to go into DNA and make very precise rewriting of the code inside of those T cells to not only put the artificial receptors on the surface, but to change their behavior so that they're more effective at clearing tumors once they find them. And that's the basis for a lot of the work in my lab, and for the basis of the work of a whole network of different labs around the world that we're teaming up with to try to solve this problem and take advantage of enormous opportunities that are emerging as biology makes these really rapid advances. We are in a golden era of biology where there's rapidly advancing ways to understand disease and rapidly advancing ways to intervene and hopefully cure disease.
What is your hot take?
On what?
On anything.
What's my hot take? My hot take is don't over-index on what people tell you to do, including me. I think that there's lots of people who are going to give you advice, and you should take it all in, and you should also be willing to look inside yourself and see what you really want to do. And I actually think that that's one of the things that's required to succeed, is being able to not be blind to other people's advice, but make sure you do what works for you in a reasoned and thoughtful, but also in a way that's going to make you feel passionate and exciting. Because if you're going to go into challenging fields, I think it requires that kind of internal motivation to really push yourself into something that's going to open up exciting opportunities.
What is your guilty pleasure?
I don't think I'm going to tell you that.
What is one moment when you realize you made the right career choice?
The moments that I remember that I know that I made the right career choice are when people in my lab surprise me, and it happens all the time. That ideas emerge from a conversation or from a student who has been thinking about something and comes up with an idea that the student, he or she may think it's crazy, but sometimes those are the crazy ideas that really move the needle. And sometimes people, just see these moments as I get to know people over decades and watch them mature as scientists. All of a sudden, they reach these levels where they surprise themselves and are able to really move fields forward. And that happens really at every stage of development in ways that continue to surprise me. And getting to be a part of that, every time I see these moments where people surprise themselves or come up with a new piece of understanding, I know that I made the right choice in my career.
What's been your most surprising finding?
That's a good question. I think when I look back, we took a pretty big risk in some ways of making a big bet. I guess in some ways I have to tell a story here that I started my lab in 2012, and 2012 was a year, and so I came to UCSF looking for exciting opportunities with some ideas, a lot of enthusiasm, but not a lot of clarity of what we were actually going to work on. And 2012 turned out to be an amazing year to be looking for projects, because 2012 was the year that the first pediatric patient was treated with CAR T cells and treated successfully, Emily Whitehead, by Carl June at Penn. It was also the year that Emmanuel Charpentier and Jennifer Doudna published the seminal CRISPR DNA editing paper in Science. And to have the surprise of thinking about, how these things might fit together, was being in the right place at the right time. I think, I would happen to be moving to San Francisco, not knowing about either of those advances, having already made the move out here, the intention to move out here. And then all of a sudden I had, through serendipity, got introduced to Jennifer Doudna. We teamed up on using CRISPR. And I guess it was, in some ways, a risky project to be moving and applying new technologies, to be able to use CRISPR directly in human immune cells. But I was excited about it, and I was at a position where taking a risk made sense, because the scientific payoffs would be enormous. And so I guess I was surprised by learning about CRISPR at that time, surprised that I got to meet Jennifer, surprised by what a great collaborator Jennifer Doudna has been, and then surprised by how well this has worked and how quickly this has gone. So those were all surprising aspects of my career that turned out to be very, very positive surprises.
As a scientist leading such a successful translational lab, what does it feel like to be on the frontier of advancement, to lack the ability to turn to books, papers, or even Google for answers? Who do you go to when you're stumped?
I think one of the things that's really important to understand about science is the team nature of it. I don't think it's a lone person sitting looking for inspiration. Occasionally these ideas come to someone when they're alone, but so often what happens when people get stumped is that we get together and we brainstorm and conversations. One of the things that sometimes helps is taking a walk, taking a break from it and coming back to it. But it's often conversations and testing ideas through conversation that work for me. I think people have different styles about what their creative process looks like. But for me, that's the most fun and generally the most productive way to tackle these moments where inevitably, when we're pushing boundaries, we will get stuck. And I think it's kind of the fun to be able to tackle these things together.
Alex, as a first year at Harvard, you were recognized as the most promising freshman. In grad school, you were a Sandler faculty fellow. And a year after grad school, you started the Marson Lab at Gladstone, and then became the director of the Gladstone Institute of Genomic Immunology. Describe to me step by step and in detail the steps you've taken to reach the level of success that you have.
I don't know how to answer that. What can I say? I think I've benefited from amazing teachers and being people who motivated me. My parents did a lot to nurture enthusiasm for for ideas and gave me a lot of freedom to get excited about things. And I guess I have some perfectionist tendencies and have also motivated myself. And I think that that's evolved a lot over time. I think that some of the things you listed are sort of external recognition that is really nice, but I think I spent a lot of time working really hard to make certain achievements. I think one thing that's evolved over time is now a lot of that is directed towards things that will actually improve patients' lives, to things to trying to use my sort of efforts towards things that will improve the scientific opportunities for my trainees, to work as a team to advance the quality of everyone's life here, and then to make discoveries that improve the world. And so I don't know all the steps that go into it. I think to the extent to which I've succeeded, it's definitely been because of having people who've really looked out for me and given me a lot of opportunity to pursue things that I'm excited about, and now I feel like I'm in a position where I get to do that for other people, and that's part of the cycle that is rewarding.
What advice would you give to the younger generation of up-and-coming scientists?
Don't do the things that are hot now. Do the things that you are long-term excited about. Don't make calculations that are strategic to try to do exactly what you see people succeeding on now. Think about the problems that are going to interest you for a long period of time, and that will evolve. You have plenty of freedom to make moves from one topic to another, more than you think. But do the things that you see as the problems that you want to solve. And go after them ambitiously. That's what science is. Science is a field where we get to be curious and to ask the questions that we care about. And asking any question and answering it is hard. So ask the questions that you really care about, that you will be exhilarated about as you learn the answers, that will make the world a better place as you learn the answers. If I had to guess, I think in the course of your lives, one place that might make a huge difference is thinking about questions that will make our planet more habitable and improve the quality of our lives in the face of a climate crisis. Think about social justice. Think about how you can make the world better, but be true to what exhilarates you as you think about that. And I guess the other piece of advice that I think is aligned with that is, please remember that there's always more possibilities than people will tell you. As much as I'm telling you, you have the freedom to explore and to define what you want your career to look like. I think it's even more than I'm saying. I'm probably understating you there. You have to find the environments where people champion you and give you the freedom to craft a career that's going to make sense for you. And I think you can find those right mentors. Talk to me, talk to other people who are committed to this idea of not reproducing what they did, but helping find ways to encourage the career that makes sense for you. And I think that's what great scientific, great academic, other types of great centers do to cultivate innovation. They encourage people to pursue the questions that are most motivating to them.
All right, everyone. Dr. Alexander Marson, thank you so much.
Thank you.